All right, welcome back. Today we're going to start talking about the process of DNA replication. And of course, DNA replication is simply the process by which genetic material is copied. So we have the original DNA strands. They are used as templates for the synthesis of new strands. It occurs very quickly. This is important because genomes can be very large and we can't spend an excessive amount of time during replication, so it needs to be quick, but at the same time, it also needs to be very accurate. We can't have mistakes being made in our genetic code that can lead to life-threatening mutations. And also, it can only occur at the appropriate time in the life of a particular cell. So this process is very controlled. It doesn't start accidentally. So our first learning outcome here is to describe the structural features of DNA that enable it to be replicated. So as you remember, DNA replication relies on the fact that we have two complementary strands of DNA. A is always base pair with T's, and G is always base pairs with C's. So in summary, we have these two complementary strands of DNA, and that DNA separates and becomes two separate strands of DNA. Each one of those will serve as a template for the synthesis of a new complementary strand. The two newly made DNA strands can be referred to as the daughter strands. The original ones can be referred to as the parental strand. So here's a schematic. Up here you can see what a parental double strand piece of DNA would look like. It's our basic double helix. And we'll color those in purple. First thing we have to do is separate the two strands. And notice we do have anti-parallel here, so 5' to 3', and 5' to 3'. So we separate, and once we've separated, we can bring in new nucleotides to base pair with appropriate. So here's a C matching with a G, C with a G. Here we'd have a T coming in, a G coming in. And we have the same thing happening down here. So here's a C, we have a G coming in, C coming in to match with a G. And this allows two evolving daughter strands to be synthesized. Over here in the schematic, you can see we have a particular piece of DNA. You can look at the sequence, CA, C, T, GG, and so forth. What we have to do is separate the two and fill in the missing base pairs on both, and you'll see that both of these are identical to the original. You'll notice over here, one of these daughter strands is referred to as the leading strand, and one is referred to as the lagging strand. And we'll go into detail why this is, but in short, DNA is always replicated in a 5' to 3' direction, the same direction that we read the DNA. And this has to do with the structure of the nucleotides themselves. And we'll go into more detail later on. Now, originally when we were investigating how DNA was replicated, there were a couple of possibilities of how this might work out. So one was called the conservative model. So here we have two parental strands in purple. We somehow replicate that DNA and we end up with the two parentals over here and the two daughters over here. We go through a second round of replication. We'd end up with one original set and then 3 copies, because this one would get copied, so it has a copy here. This one would be present down here, so this and this are the same. Here's a copy. So this is referred to as the conservative model because This would imply that there's an organism out there that has its original DNA from all the way back when. There is something called the semi-conservative model, and here we see the two parental DNA strands. They are separated and then filled in with a daughter strand that is complementary. Same over here. So this has one of the two strands being original, and this has one of the two strands being original. We go through a round of replication again, so this gets separated. Again, the strand would come down here and serve as a template for this strand. This blue strand would come down here and serve as a template. And so we would have one original strand here, and a daughter strand. These are all going to be daughter strands. This happens to be the correct model, so semi-conservative is correct. Yet another distinct possibility, just looking at the theoretical possibilities, is that you take the original DNA, in the process of replicating it, it becomes a mismatch of where the original and where the daughter DNA is. And this would be referred to as the dispersive model, because where the original chunks of DNA are would be kind of randomly scattered throughout the daughters. So the way we investigated this is we took a suspension of DNA that was labeled with a particular isotope of nitrogen. So this is nitrogen 15. This happens to be a radioactive version, or a heavier version. It essentially has an extra neutron. Here is nitrogen 14. So here are bacteria that were originally grown in the presence of only nitrogen 15. So everything inside of it is built out of nitrogen 15. We then replace the media with nitrogen 14 and follow what happens. Okay? As the bacteria grow, we will take out samples and lyse them, and then put them over a gradient, which allows us to separate N15 from N14 based upon the fact that N15 has an extra neutron and everything is going to weigh slightly more. And then we can look to see what's left over. So this is the actual experimental level. From a conceptual level, we have a bacteria that has DNA made with all nitrogen 15. We replace that with nitrogen 14. So depending on which model turns out to be true, you could have two daughter cells with only half the DNA being heavy, and down here we would have half the DNA being heavy, but the cell would have none of the DNA being heavy. So we progress this through several generations. We put this, lyse the cells and take the DNA and separate it by weight, and then we can detect. And there are some distinct possibilities. You could have all nitrogen 14 DNA or light DNA. You could have half heavy DNA, which one strand is made out of nitrogen 15 and one is made out of nitrogen 14. Or you could have fully heavy DNA, and this is what you would see in the parental strain. So here's the actual data. And across the top here, we have the number of generations that have happened. So how many times a bacteria has divided. So here it is divided once. Here it is divided twice. And here is generation 3 and 4. So this is progressively more and more. What you'll notice here is when you are at the very beginning, so at point 3, so we haven't even completed one generation yet, you'll see that most of the DNA is labeled as heavy. So this is consistent with a parental strain. After one generation, we split it, and you can see that almost all of the DNA is this half heavy. And then as we start progressing on further, we get more and more of the light DNA being present, with a half heavy being still present continually on out, although in fewer and fewer ratios. So this essentially leads us to follow along with this semi-conservative model. And logically, now that we know what's happening at the molecular level, such as here, this makes a whole lot of sense. All right, so now we are going to describe the key features of bacterial origins of replication and explain how DNA, which is the name of a protein, initiates DNA replication. So DNA from bacteria, as we've discussed before, has a single origin of replication. Remember, this is a key feature that's different in eukaryotes. Eukaryotes have several. Bacteria only have one. So if replication is starting at this point, we will get DNA strand separation, and we'll start filling in the daughter strands. And this will happen in both directions from this origin of replication. So you'll have one round of replication moving in this direction and another moving in this opposite direction. These are referred to as replication forks. This will continue to progress around until we finally reach the site where replication ends. And then at some point there, magically, the process ends by the replication forks running into each other, and then stopping, and the DNA is separated. So here you can see a capture, a photograph of this actually happening. So here is a plasmid, and it is being replicated, and you can see two replication forks extending around the circle. Now, if we look at the origin of replication and look at its actual components, it has three main types of sequences. One is an AT-rich region. So here we have regions that have excessive amounts of A's and T's. You see very few G's and C's in this region. We also have another sequence called a DNA box. You will see that this sequence is replicated over and over and over again. Not always completely perfectly, but very, very similar. The third major site is something called a GATC methylation site, and you will see a number of these scattered throughout. So, when we come back, we will walk through exactly how all of these elements lead to a replication fork forming.